Hello, good morning, everyone. Hope you had a good weekend. We have not a lot of things to do today. I just want to finish up lecture 4, and then I'm going to help you with your lab. We should be done in, an hour and a bit, and then you can go on and work on your lab, right? So yeah, let's get started. So last time, I'm just gonna do a note here for you instead of going with the PowerPoint. I think maybe that's a little bit better for some of you. Of course, I'll post a note afterwards as well. Okay, so like if you have a piece of paper, I want you to write transport here. Okay, transport right in the middle, and then I'm gonna do a flow chart that goes all the way, down all to the page. Okay, so start in the middle here, not super big, just at the top of the page if you could. And I'm gonna try to do the same thing here. Okay, we're gonna start with transport. Transport. So we can say membrane transport or transport across the membrane. Maybe that's better. Transport across cell membrane. Okay. Now we know that there are two kinds of transport, right? Two kinds of transport. Okay, one kind over there, and then we'll do the other kind over here. All right. Just gonna give you a moment to copy that down if you need to. I mean, you should copy this down. Not if you need to. Hello, for those of you who are joining us just now, we are taking a note. So please take out a piece of paper. The two type of transport is basically passive transport. Passive transport on one hand, and we have active transport on the other hand. Okay, active transport. So can someone tell me the difference between the two? We briefly talked about it last time, but anybody wanna? Okay, so use of energy, yeah, they differ depending on whether they need ATP or not, right? So for passive transport over here, no ATP is required. And in terms of the direction of the transport, it is going from high concentration to low concentration. Can you guys see my writing? Is it too small? Let me know if I need to zoom in, okay? So I know some of you are probably on your phone and I don't know, maybe too small for the phone. Okay, so this happens spontaneously on its own. No energy input from the cell is required. All right, we're gonna go to active transport in a bit, but let's break this down into a few more categories. All right. So we have three things here that I want to talk about. Right, so first one is simple diffusion. Simple diffusion. And that is movement of substance oops, I should use blue. You should also use color if you could, okay? Because I'm gonna use different colors for different things. Blue is just a description, and I'll use red for examples. Movement of substance. From high concentration to low concentration. Okay. Without any help. It just happens on its own. There's no need for support. Okay, there's no need for membrane protein to be present, for example. Okay. And simple diffusion is ideal for small non-polar, okay, molecules. Okay. Also suitable for hydrophobic molecules. To cross the cell membrane. Okay. So let's give some examples, right? Like O2, CO2, these are small non-polar molecules. You can have hydrophobic molecules like, some kind of lipids like steroids. Okay? So all these are capable of crossing the cell membrane. We have water who can do this, even though water is polar, but it's small enough. Okay, so they can either be small, okay, non-polar, or hydrophilic molecules, right? Okay, you know what? Maybe, I mean, you don't have to erase yours, okay? Like I am just gonna be moving it over here, and then small, right? Small, we can put like O2, H2, we can put H2O small. Okay, non-polar molecules like O2, CO2, and then hydrophobic molecules like steroids. Okay, these are just some examples, right? So that's simple diffusion. It's passive transport. No energy is required. And we have another kind, right? Osmosis. Osmosis is basically the diffusion of water, right? Okay. So it's still a type of diffusion, but then we specifically focusing on water, right? Diffusion of water towards the more concentrated area. Okay, so what you can think of is like think of the isotonic, hypertonic, and hypotonic cases, right? Okay. In those cases, water either move into the cell or out of the cell, depending on the concentration of the environment versus the inside, right? We did this last time, remember? Okay, so that's basically osmosis. It's a type of passive transport, focusing specifically on water alone. Okay. And we have another type here. Am I going too fast? Are you guys able to copy everything? Should I take a break? Should I pause for a second? How are we doing?
Yes, please.
Oh, it's pause? Okay, yeah, all right. I'll give you a moment. Are we okay now? Again, I'll post this
Go
out.
ahead. Thank you.
Yep, okay. Thank you. I'll post this afterwards as well, should you need sure, I can wait a little bit longer. The rest of you, while you're waiting, right? Make sure you have a copy of the lab with you, the lab report, right? Because we are going to be going through it in more detail today. I hope you have watched the video that I post with the lab. Um, Otherwise, like it wouldn't really make sense um, to go through the lab questions. All right, so the last kind is called facilitated. Facilitated diffusion. By the way, we talked about all this already last class. I'm just putting it in a chart for you. Okay, so facilitated diffusion is still under passive transport. It still does not require energy. We are still going from high concentration to low concentration. Okay, the difference is that the molecule that is trying to go through is too big. Okay, so big molecules, okay, big or charged molecules cannot cross the cell membrane themselves. Okay, they require the help of membrane proteins. Okay, we learn about membrane proteins. We know they have integral proteins and peripheral proteins, and they have different functions, right? Attachment to cytoskeleton, enzymatic activity, cell-cell signaling, right? So on and so forth. And one of the 6 functions that we talked about is transport. Okay? So in this case, the molecules are very big. They won't fit through the gaps in the membrane, or they are charged and therefore cannot go through the hydrophobic tails. So in order to cross, they would need to have the help from these membrane proteins. Okay, so there are 2 kinds of facilitated diffusion. All right. Okay, we have channel protein, and carrier protein. They are both facilitated diffusion. They are both membrane protein that helps move things across the cell membrane, but the way that they do it is a little bit different. Okay, so for channel protein, right, it's an integral protein That means it goes all the way through. That provides a tunnel for charged molecules or charged particles to pass through. Okay, so example, calcium. Okay, basically any ion that you can think of, right? And so could I scroll up? Okay, yeah, sure. There you go. Is that up enough or even higher? I don't think I can fit them into the screen. Sorry, Kiana. Maybe I'll zoom out a little bit. There. Is that okay? Oops. All right. So I'm gonna draw your picture to show you what it looks like. You must be sick of drawing these circles and lines now, but those are phospholipid bilayers, right? Okay, there you go. And we will have the channel protein right here. There's going to be a tunnel right in the middle. Okay, and then you have the charged particle. And then they will go through the tunnel. Okay? So the tunnel basically shields the charged particle from the environment, right? They don't interact well with the hydrophobic environment because they're charged, but by going through the tunnel, they are protected from it. They are shielded from it. Okay. Now, it's important to understand that the protein does not dictate the direction of the movement. The protein does not control whether the charged particle comes in or goes out. Okay, the protein is just there, providing a tunnel. Okay, what dictates the direction is the concentration gradient. If it's high concentration on the outside and low on the inside, they'll come in. If the concentration switch, it's higher on the inside and lower on the outside, then they will leave. Okay, so direction is not dictated by the protein. Okay, it's dictated by the concentration gradient. I'll write that down for you in a bit. But let me finish carrier protein first. Carrier protein is also integral protein. Integral protein that can change its shape slightly to move large molecules across the membrane. Okay. Now I'll draw it out for you. Okay, so it's like this. It faces one side. Okay, it will grab onto a big molecule, and then it will change the shape slightly, and then it will spit it out on the other side. Okay. You guys know the game like Pac-Man, right? Pac-Man, the thing that eats the dot, right? This is like that. Okay. It grabs onto it, and then it opens on the other side and spit it out. Right? And then it turns around again, grab another one, and then spit it out. Right? So I'm gonna put here the membrane protein. In this case, okay, the membrane protein do not control the direction of movement. Okay. The concentration gradient does. Okay. Let me know if that's okay. If you guys have any question, please let me know. I'll pause for a second for you to catch up. Oh yeah, I forgot to give you an example, right? Like, so what's considered as a big thing, right? Example, glucose. All right. Any question? Are we done? Copying? That's all for passive transport, guys. Okay. I can zoom out a little bit, like for if you want to take like a screen grab or something. But again, like I'll post the notes after class. But if you want, take a screenshot, take a picture. All right. So next we're gonna do active transport, and I am just gonna move it over here to have more space. I know you cannot do that on your page, but you should reorganize your notes after class anyways, right? In the process of redrawing this, you probably will be able to remember it a little bit better. Okay, so now we're gonna move on to active transport over here. So for active transport, it requires ATP, right? And we are going against the concentration gradient from low concentration to high concentration. Okay, so we can say against concentration gradient. Uphill, right? If you want, you can go back. I'll come back in a second. Over here, we can say down concentration gradient, downhill. A gradient is just a difference, right? Okay, so down concentration gradient over there, against concentration gradient for active transport. All right. Now, for those of you who are planning the space, it's gonna look something like this. Okay, so I'm gonna have one thing here, and then I'm gonna have one thing here, and then that one thing will have two things, and then one of the two thing will have three things. Okay, so that's where we're going. All right, so plan your space accordingly. Get a second piece of paper if you need to. Okay. All right. So the first thing here. First thing is called a protein pump. Protein pump. So protein pump, they basically uses ATP to transport molecules against their concentration gradient. Okay, I mean, it sounds exactly like this definition, right? But that's what it is. Okay, so you can say, I guess we can add an extra bit here. They are integral protein that uses ATP to transport molecules against their concentration gradient. So an example, the only example I'm going to give you is something called a sodium-potassium pump. You don't have to know a lot about it, but I'll tell you the basics. Okay, I'll give you a minute to finish copying. Now I'm hoping this is more clear, right, for some of you, than you know, me going through the PowerPoint slides. Like, I mean, I try to do a combination of both, right? So, you know, we cannot do everything on the note like this, because otherwise we'll run out of time, right? But you know, sometimes for complicated things, maybe this will help a little bit. All right. So let's draw a sodium-potassium pump. Now you just have to believe me, okay? Sodium-potassium pump is necessary for the normal function of cells, including things like nerve cells. Okay, we'll learn more about it next semester if you take the anatomy and physiology. Okay, so this is the inside of the cell, the cytoplasm, and this is the outside. And again, you just have to believe me. In order for this to work properly, in order for the nerve cells to function properly, you have to have high sodium on the outside and low sodium on the inside. And conversely, you need to have low potassium on the outside and high potassium on the inside. Okay. So how does the cell maintain that condition? Well, I do not want sodium on the inside of the cell, so the sodium-potassium pump will have to pump the sodium out, right? And you can see it's going from low concentration to high concentration against its concentration gradient, which is why you need to use ATP. And at the same time, we want to have more potassium on the inside, so we're bringing it in. Okay. So for this to work, again, you would need to have ATP used. Okay. So this sodium potassium pump, for every, let me just move this aside here, high sodium on the outside, right? So for this to work, sorry, I have to write here first. Okay. For the sodium potassium pump, for every ATP that it uses, it will pump 3 sodium out and it will bring in 2 potassium. Okay. So that's it. That's basically it. All right. There are other kinds of pumps. Right? Bacteria, for example, they can evolve to have an antibiotic pump. So if you try to kill the bacteria with antibiotics goes in, they will pump it out. Right? They are resistant to the antibiotics in that case. Okay, there are proton pumps in our body to create the acid in the stomach, for example. Okay, some people who have like very bad heartburn, okay, sometimes they might have to take a proton pump inhibitor, okay, PPI, right, for heartburn. Right? If you take the Tums is not enough, then you take the proton pump inhibitor. It stops your stomach from producing too much acid, basically. Okay. All right, moving on. Next, we have bulk transport. Bulk means a lot, right? Or really big. Right, so if you need to move a lot of things in and out of the cell, or if you need to move something that's like super big, you have to use bulk transport. Okay, so in terms of bulk transport, you can be moving things in, or you can be moving things out. Let's do the one that you move things out. So this is called exocytosis. Okay. Secreting, you can say secrete, yeah, secretes large molecules from the cell. Okay. I will try to draw a series of pictures to help you understand this. Okay? Don't draw this yet, just watch and see if you understand it first. Okay, so this is cell membrane. Okay. So we might have a vesicle that's like freshly made from the Golgi body, right? Okay, there's a protein in here. And I showed you before, this vesicle has multiple destinations, right? It could stay in the cytoplasm, it could become part of the membrane, or you can excrete it directly out of the cell if you need to use it, right? So that's what's going to happen. This vesicle from Golgi is going to move towards the cell membrane. Okay, and then it's going to touch the cell membrane. It's going to touch the cell membrane. Let's get closer right here. There we go. It's going to touch the cell membrane. Oops, wrong color. No, hold on. It's going to touch the cell membrane. And then is going to basically open it up a little bit like that. See if I can get my eraser here. There we go. Okay, and then finally, oh boy. And finally, it is gonna basically open up completely. Right? Okay, so this thing can come out of the cell, right? So this is called exocytosis. Okay, moving huge things out of the cell. Does it make sense? Okay, so the example is protein from vesicles. All right, I'll give you a moment to copy that down. And when you're ready, can you tell me the opposite of exocytosis? What's the opposite of exo? Anybody? Endo, that's right. Endocytosis. That's to bring things in, right? endocytosis. So this is to absorb, to bring in, I'm just gonna say bring in. Bring in large molecules bring large molecules into the cell. All right, and it depends on what you're trying to bring in. We're gonna have different names for them. Okay, let's split them up. We're almost there. Hang in there. Just need to do it for another, I say, 8 minutes, maybe 10 minutes tops. Okay. If you are bringing in solid, then we say phagocytosis. Okay, phagocytosis means cell eating. By the way, all these are active transport, right? All these requires energy, right? So this is cell eating. Okay. Brings in solid. Okay. So example, bacteria. Okay. So here you have a white blood cell. Okay. White blood cell, WBC. It's part of your immune system. And here is a bacterium. That's too big right here. So what happens is the white blood cells will go and surrounds the bacteria like that. Okay. And then it would eat it up. That's called phagocytosis. Okay. You do this every day. Okay? You have macrophage all over the body. In the lungs, for example, we breathe in a lot of, bad stuff from the air. In the lungs, you have you have these white cells that would just eat up all those things. You have white blood cells in the liver and the spleen that would eat up worn out red blood cells. Okay, your red blood cells are only good for 3 months. Afterwards, you gotta destroy them. Right? And that's what the white cells will do. They will eat them up and break it up. Quick question: does anybody remember what organelle is going to break down the bacteria now? Now that we have eaten it up using phagocytosis, what organelle will destroy the bacteria? Okay, I see Golgi body, mitochondria. Good try. Not quite though. Anyone else?
It's the lysosome.
It is the lysosome. That's right. Remember, the lysosome contains special enzymes, right? A mixture of enzymes that will break down the things that is coming into the cell, right? I hope that rings a bell. If it doesn't, that's okay. You still have time before your first quiz, before your second quiz, rather. Right. So here is the second type of endocytosis. You're bringing things in. So if you're bringing solid, we call it phagocytosis, cell eating. You can also drink a little bit. Okay, so that's called pinocytosis. This is cell drinking. That's what it means, okay? Like if you translate it. Pino is drinking, cyto is cell. And this is to bring in mostly liquid. Okay, so you're bringing a little bit of the extracellular fluid into the cell membrane, into the cell rather. So like your egg does this, like the human egg. Kidney cells sometimes do this as well. All right, so that's pinocytosis. It's the exact same thing, but rather than bringing in a chunk of thing, it's just mostly liquid. And then we have our final thing here, final type. It's called receptor mediated. I'm running out of space. Receptor mediated endocytosis. Okay. Only molecules that match a receptor can be brought in. Can be brought into the cell. Okay, so let me just draw it out to help you explain it. Okay, so look here, right? This is cell membrane. There are some receptors here. Okay. So, if I have two kinds of molecules, let's say we have the triangles here, and then we have the circles. Okay. Which one will we be able to bring in? Hopefully, it's not a very difficult question. Only the circles match the receptor, right? Match the receptor, right? Receptors. Only the circles will come in. Make sense? Triangles, if they want to come in, too bad. We don't have a receptor for it. They're not allowed to come in. Okay? So receptor mediated endocytosis. It's more selective. There is like, it's more choosy. Okay. It screens the thing, and only if you match will you be able to come in. Okay. That's it. That is all of lecture 4. Are we okay? Any question here? That's like a really big chart. Okay, I'll post this online. But do we have any questions at all? Okay. What is going on in the class? Well, I think your lab is due Thursday, and then your quiz is going to be next week, right? I forgot what day. Hold on, let me just quickly
You said after Thanksgiving?
yeah, it is after Thanksgiving, but I don't remember if it's Tuesday or Thursday. Let me just open up my schedule and see. Where is it? There we go. You guys 201. The quiz will be on Tuesday. Okay, Tuesday.
So actually like right after.
Yeah, right after Thanksgiving. You can
Literally right after.
you can give thanks that the quiz is after and not before. Okay. Now I do want to go through a few poll questions with you, because some of these will show up on your quiz. Okay. If you could go to your PowerPoints, that would be great. If you don't have your PowerPoint with you, that's okay as well. Just write down the page, the slide number, and write down the answers. Okay. So here we go. This is slide number 46. Okay, slide number 46. I'm just gonna go through them with you, okay? Because we don't have a lot of time. I want to talk about the lab and let you go early to work on the lab, okay? So passive membrane transport processes include, all right, so let's see. First one, consumption of ATP. Nope, no ATP is required for passive transport, right? We literally just took a note on that. Movement of substance down its concentration gradient. That's the one. Goes from high concentration to low concentration. Okay, let's see why the other ones are wrong. The use of transport proteins when moving substance from area of low to high. No, it's high to low, right? Movement of water from an area of low concentration to an area of high concentration. Okay, so that is not passive transport as a whole, right? This is describing osmosis specifically, right? So the best answer is B. This is slide 47. When a cell is placed in a hypertonic solution, what's going to happen? Now, before you look at the answers, right, you should look at the notes. Again, make a cheat sheet that allows you to look at the information quickly. So this is the cell. Hypertonic means they're more solid inside than outside. Okay, so sorry, the other way around. See, it's easy to get confused, right? So hypertonic is more solid outside than inside. So water will be leaving the cell. These arrows represent water. I mean, some will come in, but most will be leaving, right? So let's see which one applies. Net movement of water out of the cell. That's correct. The cell will burst. Well, if water are leaving, it's going to be shrinking, right? It's not going to be swelling up. So it will not burst, but it will shrink. A shrink is called plasmolysis. We wrote it down in the notes last time. The cell will only burst if it's an animal cell. Again, no bursting is going to be happening here. Solution concentration is higher outside compared to the inside. That is also correct. All right. So just organize the information for hypertonic, isotonic, and hypotonic in a chart or something. That way you'll be able to access the information quite easily. Okay, so that was 47. Again, if you don't have the slide, would you just write down 47 on your note, in your note, and then write ACE. So that way you will have the answers afterwards. Okay, now I don't know if you noticed, all right, but there were some questions on the first quiz that are taken from the concept checking question, right? So make sure you review these as part of your studying. All right, that's basically what we did just now. And here are more questions. Which of the following can cross the cell membrane the easiest? Okay, so let's see. Small charged molecules. Anything that is charged is going to have a hard time crossing. Okay, like, I mean, they can still cross, right? With like facilitated diffusion, for example. But the question is asking for like the easiest. Okay, like without help, can able to do it on its own. So we have large hydrophobic molecules. Anything that's hydrophobic will be able to cross. But the problem is this is large, right? Like if it's too big, it's not going to be able to do it on its own, right? You might need to have a carrier protein that does that for you. Okay. But if it's small and it's hydrophobic, that is the perfect choice. Okay. They are able to cross on their own, no problem. Hydrophilic molecules will not be able to cross on their own, okay, because of the hydrophobic tails in the cell membrane. Bacterium is not going to be able to cross easily. If you want to eat them up, you have to do. Anybody remember what was the process called? If you have to eat up a bacteria, starts with a P. Look at the notes. That's right,
Repeat
Roy.
here.
Phagocytosis. Okay. Phagocytosis. So that's that. Okay, the answer is C. This is slide number 40. Nope, I'm lying. This is slide number, I don't even know. Hold on. 60. Okay, slide 60. The answer is C. 61. Okay, now this is a long one, okay? A patient has had a serious accident and lost a lot of blood. To replenish body fluids, distilled water just means like water on its own, okay? Sterilized water on its own, equal to the volume of blood loss, is transferred directly into one of his veins. Okay, what will be the most probable result of this transfusion? Okay, poor guy's bleeding out, and then you're like, oh, he lost a lot of blood. All right, looks like he lost a liter of blood. Let's give him a liter of water. Is that gonna help him? Is that gonna make him feel better? Is that gonna have no
No.
effect on? No, what's wrong? What's gonna happen?
Blood going on.
A lot going on. Okay,
Blood
Oh.
go out.
blood go out. Okay. Jamie said swelling. Swelling of what? Can you be more specific? I think you're on the right track.
His veins.
His
They will
veins will swell. Okay. Medicine say the cells will swell. Yeah, that's basically what we're trying to get at, right? If you give just distilled water, you will create a hypotonic environment, right? For his cells, right? For his blood cells, for example, and that's not good. Right? You're diluting the blood too much. It's going to become hypotonic, and the cells will swell up, right? And then they might burst. Right? So excessive blood loss is not a good idea to give them a lot of water. Okay, if you're gonna do transfusion, you're gonna actually want to do either isotonic or even hypertonic in some cases to help retain the fluid in the blood vessel. All right, that's it. Oh, this is a very long one. I'll read it to you. 5 bags constructed from semi-permeable membrane that is impermeable to sucrose. Okay, so what does that mean, right? So we have a bag. Okay, that's gonna be semi-permeable. That means some things can go in, some things can go cannot, and it's impermeable to sucrose. It means it won't let sucrose pass through. Were filled with various concentration of sucrose solution. So in the bag, we have different concentration, okay, of sucrose, and then place in separate beakers containing initial concentration of 0.6 m. Okay, m is just the unit for measuring concentration. At 10-minute intervals, the bag were weighted and percent change in mass of each bag were graphed below. Which line represents the bag that contains the solution isotonic to 0.6 molar solution at the beginning of experiment? Okay, so isotonic to the outside, to the beaker, means it's the same concentration, right? So that means if it's 0.6 m outside, it's also 0.6 m inside. Again, you didn't really have to understand what the m represent. It's just concentration of a particular number, right? Okay, so some of you were saying, right, that the wordings were a little bit weird on the quiz, and that made it hard to understand the question. I assume something like this is what you meant, right? Like, it's so wordy, it's so much information, right? So I'm trying to help you understand how to, break down a question like this and answer it. So in this case, because it's the same on the inside and outside, and only water can move, so water will move in water will move out as well, and they cancel each other, right? And so overall, what's going to happen to the volume of the bag? Well, because it's isotonic, you should expect no change. That's why the answer is C. Okay, C here, no change. All right. A would be for like hypotonic. And then E would be like hypertonic. Now you might wonder, why is it not B? Like, how come B is not hypotonic, right? Well, in reality, the water will come in, but then the bag is not gonna keep on getting bigger and bigger and bigger and bigger and bigger, right? Like eventually the bag will not be able to hold any more water, and afterwards, right, like it would just plateau, right? It would not change anymore. Same thing, right? Water will leave, but after a while, when it's the same, it will no longer continue to leave, right? It will flatten out. So that's why like A and E are representative of hypo and hyper, but not B and E. Okay, they're not. This implies it's gonna keep on going up forever, right? Like that's not what happens in reality. All right, so if you see a question like this on a test, right, pay attention to what condition it is at. Okay, last one, I want to do a poll with you. And this question will help you answer one of the harder questions on the lab. All right. Don't choose the answer that is like the obvious one. Okay, this is a trick question. Okay, think about it and then, choose the answer. Anyone else? Only have 7, 8 responses. Okay. Let's take a look at this. After freezing and thawing some animal cells, what would happen if they are placed in a hypotonic solution? All right. Now, if you see hypotonic, right, you might think, oh, water's gonna come in, right, because of the hypotonic, and it's gonna swell and burst, right? But the trick here is freezing and thawing. Right? If you fill up a water bottle and you put it in the freezer, what's gonna happen afterwards? What happens to the water bottle after? Tell me.
Freezes, turns to ice.
Yes, it turns into ice, correct. But then, like, what about the bottle itself? Anybody? The same? Really? Shrinks? Really? You guys never done this before?
I mean, from my experience, the water bottle breaks if it's plastic.
That's right. The water bottle expands, right? Do you guys know this? Yeah, if you, that's why you cannot fill the water bottle all the way to the top and put it in the freezer, right? It will expand and pop. Like I
I thought it was
sometimes.
like after though. Isn't the question like after you thaw it?
Oh,
I
no, I'm just like I'm not talking about the question specifically. I'm just like, in general, if you put it in the freezer, I'm trying to explain the situation, right? And then I'll go back to the thawing, okay? But do we agree that if you don't know this, try it, okay? If you put water in a plastic bottle, you don't have to fill it up, okay? Like fill it up to whatever level you want, okay? Draw a line there, and then, after it freezes, you take it out, you will see that it expands. Okay. I made a mistake of, sometimes I want like cold Coca-Cola, but then like I don't have any in the fridge, so I take like one and then put it in the freezer, and then I forget to take it out, and then it just expands. Sometimes it even bursts, right? So the key thing to know here is when you freeze water, water expands, right? So in a cell, when you freeze a cell, the expansion of the water and the ice crystals that are forming will actually pierce, it will puncture the cell membrane. So afterwards, right, after you thaw it, the cell membrane is full of holes from the ice crystals. So it actually doesn't matter. It doesn't matter what you put it in after. You put it in isotonic, hypertonic, hypotonic. It doesn't matter. The water that is going in and out will just be the same because it's just full of holes now. Okay? There's no regulation. That's why the animal cells will not change size. Do you understand what I'm trying to get at here? Okay, and you have to understand this concept. You have to remember, freezing and thawing will damage the cell membrane, completely destroyed it because of the ice crystals. And then, it doesn't even matter what you put it in anymore. Okay. So usually people have a question about reproductive technology here. Anybody know what I'm talking about?
Sperm.
Okay, what about sperm?
Do you freeze them after you donate them? Because that's what I was thinking.
That's right. And then what's the question for me?
Freezing and thawing.
Okay, yeah, right. Exactly, right. Not just sperm, right? People freeze their egg as well, right? Celebrities, they do that all the time, apparently, right? They freeze their egg and then they can have a kid later on if they want, right? Preserve the quality of the egg, right? So the question people usually ask is, how can you freeze the sperm? How can you freeze the egg? Based on what I just told you, those eggs should like burst and those sperm should be destroyed from the freezing, right? Okay. So they don't, because the solution that they freeze those eggs and sperms in, they contain antifreeze. It's kind of like the windshield fluid, but like these are like non-toxic version of it, okay? So they allow the temperature to become very cold, -80 in some cases, but without forming those damaging ice crystals. And so the integrity of the cell membrane is going to be maintained. All right, so that's that. I think that's the last one. All right. Hopefully it makes sense. And the quiz will be on all of lecture 3 as well as lecture 4. All right. Now I'm gonna skip the break. I'm sorry. And I'm gonna go to the thing here now. The lab report. All right. Actually, your lab report looks a little bit different, right? This is my in-person class 1. Hold on, too many files. Where is it? Is it this one? Maybe it's this one. Give me a second. I gotta find the right file here. Beetroot lab. Beetroot lab. You know what? Maybe I'll just download it quickly. Sorry, guys. Give me a second. So how many of you have any specific questions for the lab? Now would be a good time to ask. Like I hopefully you have started on your lab report, right? Any specific questions? No? Have you guys started? I'm curious.
I thought you told us not to start yet until today because you were going to explain some stuff about it.
Oh, I said not to hand it in until after I've explained it. Yeah,
Oh
but
well, I
that's good.
started on it anyways.
Yeah, that's okay. That's good. All right. Now I gave you the purpose of the experiment last time, right? Did I not? Okay.
Yep.
So you guys can look at that again. I'll just say it verbally quickly. The purpose of the experiment is to examine the effects of temperature on membrane permeability, right? Okay, on the cell membrane permeability. So that's the one. Now, hypothesis is fine. Again, you just have to tell me how the temperature affects the membrane permeability. Not explaining it though, right? Not telling me why it's affecting it. You just have to tell me how you think is going to be affecting it. Does it make it more permeable or does it make it less permeable? That's all you have to say. How many sentences do I want? The hypothesis should just be one sentence, okay? Increasing the temperature will increase the membrane permeability or decrease the membrane permeability. That's all. Okay, keep it short. Remember, when you do these things, I want 3 C's, okay? Concise, right? Clear and correct. That's what I'm looking for, okay? Be concise means to the point, okay? Clear means like don't use fancy words for no reason, okay? Keep it straight, keep it easy to understand. And then, of course, be correct. Okay, state the dependent and the independent variable in this experiment. All right, so once again, right? Independent variable is the one that you are changing. Okay, so in this experiment, when you're watching the lady do the experiment in the video, what is she changing? That's the one you're going to put for independent variable. Okay? The dependent variable is the one that we are measuring. Okay, that we are observing. Okay, so what are we trying to observe? What are we trying to measure? That's the dependent variable. So here you can just type, you can literally type dependent variable and then whatever, and then independent variable and then whatever. Okay? Now this one people have a lot of trouble answering it. Okay. I mean, I don't know if I'm not being clear or something, but they rarely say the things I wanna hear. Okay, so this is asking you, why is beetroot chosen for this experiment? I know the color is the reason, right? Okay, so that's the easy part. Okay, a lot of people say, oh, because beetroot has a very, very prominent color. There's a, it's like bright pink or something. Okay, but why not use carrots or tomatoes? Carrot also has a bright orange color, right? Tomatoes are red, right? If you make tomato soup, it's gonna come out red. If you make carrot juice, it's gonna come out orange, right? They have pigments too. They have colors too. So I am asking you, why are we choosing beetroot and not these things? Okay. Now, some of you email me and say, well, the lady didn't explain why in the question, in the videos. I know she didn't, okay? Not all the answers are in the video. And some answers are not even on the internet. So there's no point, searching this up on Google. But think about how the experiment was done, okay? Think about if you were doing it yourself, why would using carrot be challenging? Okay, or not ideal. Okay, why would using tomatoes be a problem? Okay, think of your own answer. Right, this is what happens in the past. Like, I know you guys probably communicate with each other on like chat or something, right? So one person thought of this idea, and then everybody just copied that idea. Okay, and if it turns out wrong, then I read the same wrong description multiple times. Okay, so come up with your own idea. Right? You can probably think about why. Okay, so like just as the person is doing the beetroot, preparing the beetroot, think of what would happen if you're using carrot, if you're using tomato. Are you able to achieve the same kind of outcome as you are observing in the video? Does that make sense? I hope it makes sense. Okay, so some of you, someone messaged me and say, you know, it's due to the cell membrane. Okay, what about the cell membrane? Right, like, you got to elaborate a little bit. Okay, and that's not the reason I'm looking for, by the way. Okay, but like, you know, don't give me single words here. Like, don't be too concise in this case. Okay, elaborate. Don't assume I know what you mean. Okay. All right, safety precautions. Now, obviously, you're not doing the lab yourself, but you know, but think about it. If you have to do this in the lab, what are some safety procedures that you need to take into consideration? Okay, so do not do this. Okay, don't say, you know, wear gloves, wear lab coats, wear goggles, right? These are all the same thing. Okay, you can say all of these as like wear protective equipment, right? PPE, right? Okay, so like don't just say the same thing using different ways. You're not going to get full marks for it. Okay, so I just gave you one, right? Wear protective equipment, such as gloves and goggles, right? Okay. Does anybody want to give me another one that you want to share with the class and then use it as a class? Or like you rather keep it to yourself? It's up to you. Any other ones that you want to share? Be altruistic and help
Wear
each other out.
apron.
Wear apron, yeah, but like I said, right, wear apron is part of wearing a PPE, right? Protective equipment, right? So you
Yeah.
can say wear protective equipment such as gloves and apron. That's 1 point, right? That's what I'm saying. Okay, Madison, what do you mean by proper equipment?
Like don't use broken beakers and
Yeah,
holders.
so say it, right? So say exactly, spell it out for me, right? Okay, because if you just put proper equipment, I'll be looking at that. So like, what do you mean, right? Like, you know, like as in
Do
like,
not clean up broken glass or beacon.
yeah, so something like that, right? Osmond has another good one. Use tongs, right? Put the word use tongs for hot water test tubes, right? So things like that, right? Okay, you guys got this? Yeah. All right, all right. Again, I'm trying to help you do this. All right, don't wait until like Thursday, the 11 p.m. to do it. Do it right now after we hang up. Okay, next one, procedures. Test tubes were labeled as 20, 40, 60, 80, right? Like that. Which test tube is considered as the control? Explain what is the significance of having the control. People get this question wrong, or I should say people don't get full mark for this question for two reasons. Number one, they got the wrong control, okay? Again, because they look up the answer on the internet. People put like the strangest things. They're like, control is the tube with alcohol. And I'm like, what alcohol? There's no alcohol in this experiment, right? And again, the reason is because they did not watch the video. They just Google the answer. So don't Google the answer, okay? Watch the video and find out what the control is. The control is one of these tubes, right? It's either the 20, the 40, the 60, or the 80. So you have to find out which one is the control. That's 1 mark. The second mark is you have to explain the significance of having a control. Why do we even need a control in an experiment? Why can't we just have the experiment, right? Think about like doing vaccines testing. Why do we have to have people who don't receive the vaccines? Why not just give everybody the vaccines and see what happens? What's the purpose of having a control? Okay, believe it or not, we wrote this down during lecture 1. So if you're not sure, go back and look at it. Okay, so identify the control of this experiment. You should say the control of the experiment was the, and then tell me which tube. Is it 20? Is it 40? Is it 60? Is it 80? Which one is it? And then the purpose of having a control is so that we can blah, 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 blah. All right. It's okay if you don't get everything I'm saying right now. You can watch the recording after and you know, listen to it again. So 3 to 4 sentences. What needs to be taken into consideration when cutting the beetroot? Explain. I am not talking about not cutting your fingers here. Okay, so people say, oh, you need to be careful with the knives, okay, and not cut yourself. That is totally what this question is asking. This question is asking about how you are cutting the beetroot. How should you be cutting it in a way that assures your experiment is done in a proper manner? Okay. For example, I mean, you can watch the video and see how she cuts it, but like I don't want some pieces to be too big. I don't want some pieces to be too small. Okay, why not? Okay, how is that going to affect the experiment? Right. So you need to say what needs to be taken into consideration when cutting the beetroots. So you should make sure that the beetroots are blah 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 blah, and then you explain. If it's not prepared this way, right, the results could be affected because blah, 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 blah. Any questions so far? Am I making sense here? Do you know what you have to do? Okay, I got one yes. I'll take that as a win. Two yes, great. Okay, all right, let's move on. Why is it necessary to rinse the cut beetroots afterwards? Again, this is 2 marks, right? You will see in the experiment after she cuts it, she has to rinse it and then dry them. Right? This is important, right? Because if like you want to tell me why this is necessary. You want to tell me what happens if you don't do that. Okay, if you don't rinse them and then you continue with the experiment, how is that going to affect the results? Okay, 3 to 4 sentences. Again, you don't have to write too much. If you are to the point, you might even be able to answer this in 2 sentences. All right. Why is it important to leave the beetroots in the test tube submerged in the respective water bath for 20 minutes? Okay, so I know, again, in the video, the lady did not explain why we're doing it for 20 minutes. Okay, but I want you to think about what happens if you leave it in for just 5 minutes, okay? Or if I leave it in for like 30 minutes, for an hour, how will that
come off?
I'm sorry, what? Okay, so like how would that change the result? Okay, so in answering all these questions, I want you to explain to me if you don't do something, right, or if you do something in a different way, how will the results be affected? Okay. So like for those of you who don't know, right, like we're trying to put the beetroot that has been exposed to different temperature into water, and then we'll see how much pigment is leaking out, right? So what happens if you leave it in for only 5 minutes? What happens if you leave it in for like way too long? Okay, how will that affect the results? That's what I'm
Because
asking.
the process needed time to go
Mhm.
in and outside.
Yeah, so I think you're
But the
on to the
broken
right
off,
track, right? You're definitely on the right track, right? So just think about how you would phrase that, right? And I think you said something about needed time to blah 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 blah. So that explains why not shorter, right? Why not shorter? But what happens if you do it for too long, right? So you have to think about both cases. All right. And again, it's not something you can look up online. Just think about it, right? Just, use common sense. Describe next one. Describe the correlation you observed between the temperature and the color intensity. Again, some people get really confused what I'm asking here. Correlation just means relationship between two things. So over here, I'm literally asking you to look at the picture and tell me as the temperature increases, what is happening to the color intensity? Okay, describe it. Okay, so you would say when it gets harder, the color intensity, does it go up? Does it go down? Does it not change? That's all I need to see.
So if I say when the temperature reach at least 80 degrees, the beetroot give off more pigment, red pigment.
So I'm not specifically looking for 80 degrees, right? I'm looking for like a general relationship, right? So again, you would say something like, as the temperature increases, comma, the color intensity, and then you fill in the rest.
The
Okay.
beetroot pigments got more intense, right?
If
Would that
that's
be
what you
right?
think, that's what you can write. Okay, but that's all I can tell you. All right. As the temperature
Oh.
increases, the color intensity, and you fill in the rest. And then don't forget part B. The answer you gave in part A is an example of qualitative analysis. Explain what this means. Again, you will not believe it, but a lot of students just Google qualitative analysis and paste the answer into the box. That's not what I'm asking you to do. We talked about qualitative analysis, right? We did some exercise on it, right? So just go back to lecture 1, refresh yourself on the definition of qualitative analysis, and then explain to me what it means. Okay, that's it. Okay, don't look it up in Google. Okay, I will know if you look it up on Google. Okay, because it will sound weird, and then there are like strange words that are not supposed to be there. Next one. In this experiment, how will the results be measured? Sorry, be measured quantitatively. Okay, so this is an example of qualitative, but how can we do it quantitatively? If you watch the lady do the demo, she will show you how to do it quantitatively. Okay, so you just have to tell me what she did. That's all. Now, next one. Describe the relationship between absorbance measured by a spectral photometer and the color intensity of the solution. Okay. In the video, she actually does not use the word spectral photometer. She used the word colorimeter. Okay, let's spell out for you. Okay. Sorry, I gotta update these things later, but
Is that the one she has with the green dot? The green light?
yeah, exactly. Okay, so she called it the colorimeter. Okay, that's a lot of people say color emitter. It's a colorimeter. Okay, that's what it's called. So I will briefly explain to you how that works. Hopefully, it will help you understand better. Okay, now very quickly, a little bit of physics here. There are 3 primary colors. Okay, red. What's the other one?
Green and yellow?
Yeah, red, green, and what's the last one? Not yellow. Someone else? Anyone else?
Black?
Blue. That's right. Okay, red,
Oh.
green, and blue.
Really?
Yeah, really. You remember those old school TV that we used to have with the, the gigantic ones, right? With the cathode ray tube at the back. If you wipe those screen with a wet piece of cloth, you can see the pixels. Remember? They
Oh,
used to be.
I still have those.
Is that good
I
for
still
you, right?
have those.
You can use those to play like vintage video games, right? So those are the 3 primary colors, right? The red, green, and blue.
Isn't it
RGB.
yellow? Isn't it red, green, and yellow?
No,
No.
it's red, green, and blue.
Yeah, it's red, green, and blue. So
Red, blue, and I thought it was. I'm not green. I mean, I thought it was yellow instead of green.
No, I know, right? But it's not, right? Okay, I don't know how to convince you guys otherwise, but give me a second, okay? Let me show you.
If you guys have heard of RGB, then that's red, green, blue.
Exactly, right? RGB, right?
Those are the primary colors used to make other colors.
That's right. But that's okay if you're not convinced, no worries. I'm gonna convince you right now. Okay. This circle is blue.
But like,
And
I'm pretty sure that it's red, yellow, and blue, and then green is a secondary color. No?
I think you're talking
Because
about
like blue, because we put yellow and blue to make green.
okay, no, okay, I see where the confusion is. Okay. Just hold that thought. I'll explain it to you. I know
Okay.
why. Okay. More colors
I
here, right?
understand what she's saying. That's
Okay.
what I thought.
Look here, right? Look, first of all, right? Do you guys see the screen? Red, green, and blue, right? Okay. Let's crank up red all the way to 200. Let's crank up green all the way to 200, and then I'm gonna make blue 0. What color do you think we're gonna get? 1, 2, 3. Oh my goodness, yellow. Wow, right? And you're like, wait, what? This is not true, because you were saying like red and so what did you say? Blue and yellow makes green, right? Okay, like those Ziploc things, right? Okay, so here's the confusion. What we're talking about here is light color, okay, light waves. What you're talking about is pigments, okay? So those follow separate rules, Okay, just like when we mix green and red on like painting, we get brown, right? Okay, but if we mix green and red here, as you can see, we get yellow, right? Does that make any sense at all? Or
Another more confusing part is that if you crank up red, green, and blue, you get white.
that's right. If you make up all three of them together, then you get, you know, kind of grayish white. I think it's just the intensity that I'm having here.
I think the max
There we
is
go.
255. Yeah.
Yeah, okay, there you go, right? Does it make any sense? Right? Let's take a look at another one. All right. I promise this has something to do with the lab. Okay. Let's crank up blue all the way to 225, and then we're gonna make green 0. And we get magenta, right? So the primary light color are red, green, and blue. Have I convinced you guys? Like any follow-up questions?
I'm convinced. I've been using.
But you've been convinced since the beginning, right?
Yeah.
But what about anyone else? Who was asking me about the other stuff, right? Is this okay now?
Yes, it's good now.
Yeah. Okay, all right. So, you know, the next thing is really gonna blow your mind as well. Now that we know yellow is a combination of red and green, right? Okay. And white is all three colors. Okay, white is red, green, and blue together. So when we shine light at a yellow object, maybe I should go back to here. When we shine light at a yellow object, give me a second. Yellow object. Okay, so we can shine white light at it. White light. Okay. What color is absorbed by the yellow circle here? Which of these three colors are absorbed by the yellow circle? Justine is not allowed to answer because he might know the answer. Okay, Madeline said blue. Kiana said blue. What about the rest? Does anybody think
green.
it's not blue? Georgia, what you say?
Green.
Green. Okay. Anyone else? Okay. Well, again, think about what makes yellow, right? Yellow is green plus red, right? So in order for you to see yellow, it actually reflects the red and it reflects the green. So when the red and the green goes into your eyes, you see yellow, okay? So what is being absorbed? This one absorbs blue light, okay? Absorbs blue light. All right, let's move on. You have your test tube here. It has a pinkish color because of the beetroot, okay? Pinkish red. We'll just say red to make it easier for you to understand this. So I want to ask you, what kind of light will a red color absorb? Are you guys gonna help me out here? Okay, let's do a poll. Let's do a poll. Okay. You can choose more than one answer here. Okay. I am asking you, what color does a red tube absorb? Okay. A, B, C. A is red, B is green, C is blue. What will a red color tube absorb? Come on, you guys gotta tell me. I need to know that you know this so we can go on and you know, hopefully you do the lab correctly. Okay, well, I am glad of all the people who've participated so far. Nobody chose red. That's good. That's a good sign, right? Why is this red? This is red because it's giving off red, right? It's like reflecting off red, and then it goes into your eyes, and then you see it as red, right? Okay, so this absorbs green light and it absorbs blue light, which means when you have a colorimeter, okay, and then it shines green light at it, okay, it will absorb the green light, right? It will absorb the green light. So we call that absorbance. Absorbance. Basically, the key thing that you have to understand here is the redder it is, I don't know if that's a word, but like the more red the color is, the more green light is able to absorb. That's basically what happens, right? So why not use blue light? Can we do the experiment with blue light? Yeah, you actually can. You can use blue light as well. But I don't know if you guys are aware, blue is closer to purple, right? On the rainbow, blue is closer to purple. And purple, we say violet, right? The color violet. And ultraviolet, as you know, UV is very damaging, right? Okay, so blue light is actually much higher in energy and it's much more damaging. But it's not the damage that we worry about. It's like, it's too high energy. It's like not easy to generate. So if you want to get like a something that gives off blue light rather than green light, it's more expensive, basically. So for economic reason, we use the green light. Okay, so you shine the instrument shines a green light at the tube, and then it measures how much green light is being absorbed. So basically, the more color it has, the more green light it will absorb, the higher the absorbance. Okay, so higher absorbance means higher color intensity. Okay, and it will actually give you a number. It will say, 20% absorbance, right? 80% absorbance. If it has 80% absorbance, it means 80% of the light will be absorbed. Okay, 20% absorbance means only 20% of the light will be absorbed. Any question about this absorbance business? Okay, I hope I wasn't causing any confusion.
Sir,
Yes,
today we can hand this in?
if you want, you can hand it in today, but it's not due until Thursday, okay?
Oh, okay.
Yeah.
And we don't have a quiz coming up yet.
Quiz is next Tuesday. Yeah.
Okay.
Quiz is next Tuesday. All right. Let's see what else I can help you with here. So I just described the relationship for you. All right. You should be able to do it. And then there is the data here that you need to graph. I created a video that shows you how to do the graphing on Google Sheets. I will post that after class. You can just watch it. It's super easy. Okay. It will literally take you 10 minutes maybe. So you will have the graph and then you will paste your graph here. All right, data analysis. I'm sorry, I planned to finish this in 1 hour, but it looks like I'm talking too much again. We're gonna have to go the whole way. Okay, here. At what temperature does the absorbance peak? All right, so I want to show you a graph here. Okay. So if you have a graph that looks something like this, okay, where is the peak? Is it at A, B, C, or D? Where is the peak? Okay, Medicine says C, Osman says C. Anyone else? Anyone else saying it's
C.
something? Okay, C, Georgia, C. Okay, everybody says C. Okay, good. Okay, it's not D, right? Okay, because after C, it no longer changes, right? I mean, it might go up a little bit, but then it's pretty much flat after that, right? So the peak occurs over here. Peak. Keep that in mind when you're answering the question. All right, moving on. Do you expect the temperature where absorbance peaks would be same for membranes of other plants? So like if we don't use beetroot, if we use another thing, if we use the cabbage or something, and then you do the experiment, do you expect the peak to be at the same temperature?
No.
Okay, and then you will have to explain why. Okay, don't just put no, and then
Because the beetroot has
you sure you want to discuss it now?
Okay,
no. Okay. Right. So like you might want to keep the answer to yourself. but you want to give me a reason, okay? And I actually explained this a little bit at the beginning of lecture 4. All right, when we did the percent of saturated and saturated fat stuff. Okay, so you might want to go back there and take a look at it. Next one, provide 2 reasons that explain how temperature affects membrane permeability. She actually explained it in the video towards the end, okay? And she gave 2 distinct reasons. Make sure you include both, okay? She said it quite clearly in the video. So just watch that. Okay, what is the conclusion? Okay, again, you don't have to write too complicated stuff here. The conclusion is this. Okay, after doing the experiment, was your hypothesis correct or not correct? Like if you say temperature increases permeability, is that what happened in the experiment? Right. So you say that. Yes, increasing temperature will increase permeability, or maybe increasing temperature will decrease permeability, right? In which case you will have to reject your hypothesis. Okay, so just write a sentence about the findings of the lab and then tell us whether we should accept or reject the hypothesis. Now, it used to be like, nobody ever get this question right. Number 4, a piece of frozen beetroot is placed into a test tube of tap water at room temperature. The absorbance recorded after 20 minutes is 84. Provide an explanation for these observations. Okay, so you want to go back to this chart here. Okay, so 84 is like, very similar to these high temperatures. But over here, you freeze the thing, right? It's like from a cold a temperature. Why would the absorbance be similar to one of the higher temperature ones? Okay, so you got to think about that, and hopefully our discussion here on this last slide gives you some idea. Okay, and that's it. Honestly, it's not a difficult lab. I strongly encourage you to work on it right now while it's still fresh in your mind. If you're really stuck on something, feel free to email me. But don't email me answers and ask me to tell you if it's okay or not. Okay, I will not be answering those questions. Okay, but if you need clarification on what exactly the question is asking, then feel free to email me. Okay, I hope this helps. Okay, I hope you're gonna get 90 on this. All right, no class on Thursday. Enjoy your Thanksgiving, and then Tuesday we're gonna have the quiz. Okay, all right, that's it. Thank you for coming. I'll stick around for a few more minutes in case people have specific questions. Otherwise, that's it.